Good afternoon, everybody. Welcome to the Great Lakes Data Sharing Webinar, which is jointly hosted by the Lake Erie and Aquatic Research Network, or LEARN, and Michigan and Ohio Sea Grant. My name is George Bullerjohn. I'm the current president of LEARN, and I'm a professor at Bowling Green State University. As the president of LEARN, I encourage all who are not members to join. So please consider us when you think about networking with folks in the region. And I just like to take before I introduce our first speaker, I just like to explain really how we got here to have this webinar today. And so this emerged from a discussion at our annual LEARN meeting last November. We had a breakout session on identifying pressing issues that can be addressed by collaboration. And in our breakout group, Tom Bridgman from University of Toledo spoke up and said, there are a whole lot of data out there, and how do we know what's out there and how to access it all? What are all the databases and how best to use the massive amount of data that is accumulated over decades? And so there's so much work going on in the Great Lakes and so many major repositories, about 20 or so, to put those data into, that we wanted to emphasize the importance of using regionally focused repositories so that all of our work can inform Great Lakes models that pull data from these. As a result of our discussion, thanks to Sylvia, she made some phone calls and emails. We now have this webinar as an outcome of our LEARN meeting. And I hope this isn't a one-off event. I'd like to see this as a series of discussions on how to share, access, and manage different data types from meteorological, chemical, biological, genomic information, et cetera. So I'm not an expert on databases and data curation. I'm just a user. So I look forward to the discussion and how I may access the data that are out there. So we have 3 databases on the agenda today. We'll first start with Jennifer from GLOS. Then Anders describing GLAMR, and then the Great Lakes Commission HABS Mapper by Connor. And so first up is Jennifer from GLOS.
Thanks very much, everybody. I'm Jennifer Bamey. I'm here with the Great Lakes Observing System. I've been CEO since August 2023, and I'm excited to be here today to tell you a little bit more about GLOS and how we've situated ourselves here in the Great Lakes data environment. So the Great Lakes Observing System is a nonprofit that provides end-to-end data services in support of science policy, management, industry. We work in both the US and Canada. So whether you're deciding to leave the dock on your boat or you're a manager trying to address the effects of climate change and manage flooding in your community, we want to equip observers with high quality, up-to-date lake information from the region's observing network. We are a part of the International Ocean Observing System. However, we are independent. We operate with an obligation to aggregate and share data and information about the Great Lakes. One of GLOS's goals is to be the primary access point for this type of information for the region and beyond. GLOS is fundamentally an organization that's connected through a network of partnerships and relationships and data providers. Everything GLOS does is centered around this tenet of accessibility, availability, and the sharing of data and information in the Great Lakes. When it comes to data and information, GLOS sits at the heart of the Great Lakes, not just in geography. There are 3 primary portfolios at GLOS. The first is Smart Great Lakes. In 2019, a group of organizations from the US and Canada formed the Smart Great Lakes Initiative around common goals and priorities for improving the way a diverse region learns about and responds to lake events and informing critical policy. We also aim to play a role in directing the future of science and innovation. The initiative partners that we have so far are varied. To name a few, we have the Cleveland Water Alliance, University of Windsor, Northwestern University, and Fisheries Oceans Canada. The second portfolio I'm going to talk about is LakeBed 2030. LakeBed 2030 is an initiative to explore and map the Great Lakes to modern standards with high density data. GLOS is a leader in this area, and we help to coordinate mapping activities and lead the regional crowdsourced bathymetry program along with Orange Forest Marine. We promote advocacy and awareness not only of the need for fully mapping the Great Lakes for the first time, but also new technologies and approaches that are available to achieve that goal. And finally, we have Seagull, our powerful cloud-based platform. This is the newest released marine Internet of Things platform that serves data and information in the Great Lakes. This has an eye towards simplistic usability and preserves behind-the-scenes complexity at the same time. Seagull underpins nearly everything that GLOS does with respect to data and information, throughput, visualization, and sharing. So when we think about the Great Lakes, this is a vast area. The size of the lakes and the density and volumes of people that are served, it's surprising how little we really know about them. Whether it's underwater topography, the water column itself, the biology, benthic habitat, and even just shipwrecks, we have a need to understand them. It's of paramount importance, especially when we're starting to see the impacts of climate change and how coastal resilience is affected, in particular, the effect of these on underserved communities. Understanding the marine environment, especially as we contrast that to levels of understanding that we have of the land around them and of our salty coasts, this is going to be critical for the Great Lakes region going forward. Without this understanding, we aren't going to be able to protect this fragile ecosystem as well as we might have. When GLOS looked at how we could better support the overall health of this ecosystem, we put it in a context for the data to information transformation life cycle for humans and non-humans around the lakes. We started with platforms, assets, and sensors that are deployed in the region. Data is observed, collected in situ, and automatically transmitted to cloud-based infrastructure where it's post-processed. Metadata is attributed and made ready for consumption, connection, and data sharing. We have a human-centered design applications to enable users from a variety of communities to discover data and information for a wide range of purposes, whether it's research, recreation, water quality, if you're interested in biodiversity, benthic habitats, and a variety of other interests. The last phase of the data to information lifecycle is those same users then engaging with the data and informing others to do even more with it, whether that's sharing, pulling it into new communities and users, and informing decision-making for acquisition and deploying new assets. That's all part of the ecosystem that we are trying to build and continue to support. So in 2020, we embarked on an ambitious program to develop a marine Internet of Things information technology platform that had at its core data, but with user and information as central to an approach for consumption and sharing. That result is Seagull, which is a platform to serve users, communities, partners, as well as data. So this was released in 2022, and here you can see a picture of the Seagull dashboard. It supports all the major program areas of GLOS and is aligned with International Ocean Observing System values. We aspire to be the de facto discovery utility in the Great Lakes region for data and information related to our freshwater ecosystem. This is presented in a very simple and elegant manner. However, it is powerful. It's API-driven in the cloud. It connects with a wide range of data sources that makes it available for consumption via web, mobile, and custom applications. Right on the front page, you can see the search function. Search is fundamental to the application, and it mimics our well-established patterns with machines, operating systems, and applications in other parts of our lives. It's not any different than when it comes to marine environmental data, especially data that's really critical to human and ecosystem health in the region. So as I said, Seagull's a discovery and information platform that allows anyone to learn more about the Great Lakes aquatic environment. Because it's a platform, it also allows extensions and opportunities for others to build onto it and leverage the modular backend design. A recent example of this is our partnership with Sigler to develop and maintain new features for ingesting, managing, visualizing, and sharing omics-related data. Omics refers to direct characterization and analysis of sampled pools of environmental biomolecules, particularly DNA, RNA, proteins, and metabolites. These techniques can be used to examine chemical toxicity and enable risk assessment. And so we have partnered with Sigler and their geomicrobiology lab to provide input for omics data into Siegel. This is an example of the type of information that's presented. You have an opportunity here to look at relative abundance from the sensors themselves, and also overlay with a different variety of data available in terms of abundance as well as species level organization. There's also the opportunity to compare it to the other available environmental parameters that may have been collected at a particular station. So data sharing is fundamental to GLOS's identity and mission as the certified IUCN Regional Association in the Great Lakes. GLOS's philosophy is to provide data and information services to others, but also to develop partnerships, cultivate relationships with new communities, and be a leader for observing and data-related technologies in the region. Some of the ways that we are planning to support these growth efforts include the improved new discovery tools, improving our search function on Siegel, looking for new ways to externally communicate about data and findings such as social sharing, new data entry ports that remove barriers, but also preserve integrity, privacy, and cultural sensitivities. This is important, especially as we seek to increase our engagement with tribal communities and First Nation communities around the region. We are hoping, as part of these efforts, to bring in new communities of users, including those from underserved communities or what is known as frontline communities. We're going to build new data pipelines with new data structures, data sets, and data warehouses. And we're also looking at new avenues for accessibility, including mobile application development. So if you have any questions, I am happy to answer them. This is our contact information if you would like to reach out to us directly. I'm also happy to report that Tim Kearns, our Chief Information Officer, is also online with us. So if there are any questions for GLOS, we are happy to answer. Thank you.
Great. Thanks very much, Jennifer.
As a reminder, you're welcome to use the Q&A box at the bottom of your Zoom screen. That's the easiest way to make sure that we can see your questions. And I see we have a handful. Let's see, does GLOS also collect stream data, or is it just for lakes?
So at this point, we are somewhat, we are lake focused. However, there's been discussion within the International Ocean Observing System, the IUCN regions, about the need to turn inland and make sure that we are incorporating important data from the watersheds, as well as being specifically focused on the coast. And so for us to do that, it would be important to have additional partnerships with organizations that do measure already in those areas, such as the U.S. Geological Survey. So that's something I think we're looking forward to in the future.
Thank you. And so I'm not just a disembodied voice for attendees on the webinar. This is Geneva Langland. I'm Michigan Sea Grant's communications editor, and I'm helping moderate the Q&A. We have another question. How far back does the data in GLOS go, and is historical data being added?
So I'm going to turn that over to Tim, if you can unmute yourself to help me answer this question. GLOS does currently focus on real-time data. However, we do have some of our data sets that provide longer time periods of coverage. Tim, could you speak to that?
That's a great question. So the historical data sets is available on GLOS. The availability of that data depends on the platform and what we've added so far. So we've been going through systematically to add legacy data based on different researchers and different platforms. In some cases, it goes back a couple of years. In other cases, it goes back a decade. So it really depends on the data type.
Thank you. Let's see, how does GLOS collect data from the US EPA as well as from NOAA and other Sigler research?
So I think that's a GLOS used to have a closer relationship with EPA, and I think that's something that we're looking forward to rekindling. There's, as you might imagine, lots of information at EPA, and so part of the question is understanding from EPA's perspective what data they would like to share publicly and let us house. You can think of a lot of data streams that are used to support different reporting requirements around the region, like the state of the Great Lakes reporting. There's an awful lot of information that's been collected historically, and it would be really wonderful to talk about opportunities to perhaps start serving some of that up for the public access as well. And I see Tim's hand is up.
That is a super question, and thanks, Jen, for that answer. For the person who asked it and for others, Jennifer did mention, I just want to underscore that Seagull is an API-driven platform, which means we can not only pull metadata and or data into Seagull, but we can also allow others to push content to Seagull. So GLOS doesn't need to really bear the burden or responsibility of writing all these connectors to other data warehouses and repositories. We've got the mechanism in place because it's a modular platform. And that's the difference between a platform and a product, is it allows others to develop on top of it. And we also aren't aspiring, Jennifer said it really well, which is we want to be the Discover utility in the Great Lakes. We don't want to be the data warehouse for the Great Lakes. That would be way too impractical and costly. There's lots of other data warehouses that are out there. We would aspire to be able to direct people to those data warehouses through Seagull.
Thanks. We'll take a couple more questions now, and then maybe we'll circle back at the end of the webinar if there are additional questions specifically for GLOS. But in the meantime, what do partnerships with Canadian monitoring agencies look like? Are there formal agreements or informal agreements to collect Canadian data?
Well, Canadian data is also available publicly, and we do not have formal memorandums of understanding with any Canadian agencies at the moment. I will say that, as mentioned, Fisheries Oceans Canada is participating with our Smart Great Lakes community, and so a lot of partnership development is happening through those types of initiatives. Ultimately, there are also, opportunities. They have their own infrastructure. For instance, if you're interested, someone asked a question about river and stream data. GLOS does have a partnership with DataStream in Canada where you can access this type of data. They also pull in a lot of community science-oriented data sets. So not every partnership that we have with Canada is actually agency-oriented. There are other sources of data in Canada that we are looking to grow and serve up on Seagull.
Great, thank you.
Terrific. Thanks so much, Jennifer. And next up, we have Anders Kildahl, who will be talking about GLAMR. And please tell us about all the omics going on in the Great Lakes.
Great. Thanks, George. Hi, everyone. Thanks for joining today. My name is Anders Kildahl, and I'm an assistant research scientist in the Earth and Environmental Sciences Department at the University of Michigan. Today, I'm excited to tell you about a project I started during a postdoc in Greg Dick's lab in the same department. There are many people who have really contributed to GLAMR and made it possible. So first, I'd like to highlight Greg. He's the leader of the Geomicrobiology Lab at the University of Michigan and the director of SIGLR. We also have many other people who helped to make this all possible. I'd highlight in particular Robert Hein, our bioinformatics specialist, and some other members of the team who've made it possible, especially those who've curated some of this data. But as you'll see, this is also really a collaborative effort. There are many people who have produced the data sets that go into GLAMR. So I'd highlight basically all of the omics producing Great Lakes researchers who have shared their data publicly so that we can incorporate it. And then also various funding agencies and other groups who have supported the development of GLAMR. So we keep using this term omics, but right, just to understand that a little bit better and to understand the need for this database. When we say omics techniques, we're referring to these high throughput technologies that allow us to characterize and quantify directly particular pools of macromolecules in the environment. So these can include DNA or RNA. So that would tell us who is present, or even characteristics of their genomes, right? So what genes they have, proteins or metabolites. So these molecules have a wealth of information about who is present, the genes they have, the genes they're expressing. And from these, we can infer, for instance, stressors that are being experienced or compounds that are being produced. And so this technology has really improved over the last 20 years and ushered in new and powerful ways of studying environments and ecosystems. There's a particular interest in the Great Lakes for using these technologies to monitor and better understand harmful algal blooms, especially those that happen in the western basin of Lake Erie. So while these techniques have had a significant impact on improving our understanding of the biology, they also hold a lot of potential information for modeling and forecasting and informing future management practices as well. Okay, so we know that there's a lot of public omics data out there. So basically researchers, as part of their studies, collect this omics data and they put it into existing public repositories that are really, as Tim or others were referring to them, data warehouses, right? A place to dump the data, but it's hard to actually analyze it or do much with it there. So we see that there are these entries in this particular database, NCBI SRA, where people have uploaded their data, and we see that the number of samples is increasing over time. However, these data sets are often uploaded with not very much metadata, so it can be really hard to interpret even where they came from or to compare between them. For instance, people don't always use consistent latitude and longitude, and sometimes there's not even a date associated with the sample. Other times there is much more information, but it's not all that standardized. So one of the things that we're doing with GLAMR is to really standardize this data, bring together the Great Lakes omics data, unite it and standardize it. So particularly when I'm saying standardize, I mean the metadata associated with the omics data. So this can include environmental parameters measured alongside the omics data collection, nutrient measurements, HABs relevant parameters like phycocyanin, microcystin, or others, and sample modifications or amendments. So with GLAMR, we then produce standardized omics outputs. So we take the direct characterization of these molecules and turn it into the what does this actually mean? So what are the abundance of species or the genes that we're seeing? So we take all these diverse data sets and really bring them together in a unified sense to facilitate comparisons across data sets and integration with the metadata about the samples. And I'll also highlight that GLAMR is specifically made for the Great Lakes. There are some other initiatives working to improve omics data analysis and standardization. But because we've made this targeted database for the Great Lakes, it allows us to curate it much more, optimize the metadata, and incorporate some targeted analyses, such as looking for the particular genes associated with producing toxins from harmful algal blooms, or searching for invasive species specific to the Great Lakes. So I mentioned we standardize the omics data outputs using some in-house bioinformatics pipelines. So basically we're taking this raw data and reprocessing all of it ourselves to create these outputs that are more directly comparable than if you were to take the products that each lab produced as part of its studies. So, right, we're basically taking this data that people have put into public repositories, processing it ourselves, standardizing the metadata, putting it into our backend database, and making it available via our website. So if you were to navigate to greatlakesomics.org, you would see the GLAMR website. And here you can explore various omics studies. So if you, for instance, click on this study, you can see the samples that it contains. You could look at more information about the samples. So you see this is a metagenome, basically sequencing the complete pool of DNA molecules. There are other studies that only sequence single genes, which really would only then let you see who is present. These metagenomes let you see who's present and what their genomes contain. But if you want to break samples out of the kind of confines of the original studies to really fully leverage all the data that's out there, we can also look for particular samples. So say you wanted to find metagenomes from Lake Erie, you could look at this summary table here and identify all of these samples, the data sets that they came from, associated publications, to maybe understand what others had interpreted from the data sets. We can also, though, directly analyze aspects of the data set. So for instance, if you want to see where Microcystis, which is the primary cyanobacteria responsible for the harmful algal blooms in Western Lake Erie is found, and the samples where it's the most abundant, we can search for that and see a summary of the abundance and the samples where it's observed. And so this TPM column here is a measure of the abundance of this particular organism. You could also, for instance, search for one of the genes responsible for producing microcystin, which is the toxin of most concern for these harmful algal blooms in Western Lake Erie. And if we search for that and then look at associated protein records, we can then see samples where this particular gene has been observed and the abundance of this gene as well. And so this is just one example, but using GLAMR, you could search for any taxa you're interested in or any particular function you're interested in. So we're always looking for new data submissions or feedback about GLAMR and the types of functionality that you would like to see. So if you have data you'd like to submit, please reach out to us. There's information on the GLAMR website, so greatlakesomics.org/contact, or you can email us at glamoromics@umich.edu. So with that, thank you again for attending, and be happy to answer questions.
Thanks, Anders. That's wonderful. So please put questions in the Q&A and we can continue with the discussion.
As we wait for questions to come in the Q&A box, I just wanted to share one thing that popped into the chat. Someone says, wow, GLAMR seems really cool. So it's hitting the right audience here.
Yeah, we think it's really cool too.
One participant is wondering, has the focus been on eDNA data, or are you looking for specific species sequence data as well?
Yeah, that's a good question. We've been collecting primarily the three kind of sample types you can see here. So amplicon data, and that's where I would categorize a lot of the eDNA data. So sequencing of particular marker genes, so 16S rRNA or 18S or some other markers. We have been collecting that data. At the moment, it is not, you can't really dig into the amplicon data yet on GLAMR. That is something we're actively working on. We have focused, I would say, primarily on the metagenomic data. So the sequencing of the essentially complete pool of DNA from the samples, or sampling from the complete pool anyway. But we also have metatranscriptomic data as well. So you can see some of those samples, and we can point you to where you could download that information. And in the near future, you'll be able to see summaries of the community from the eDNA data.
Great, thank you. Can you search by genes for species by name? Like, could you search for individual invasive carp subspecies?
So that would, I guess, depend on the particular species of interest. So I'm not completely sure at what similarity levels the different subspecies of invasive carp have. You can, for instance, though, search by unique sequences. So kind of one of the underlying databases we use is the UNIREF 100 database, which kind of dereplicates by exact variants. So if you could find a UNIREF 100 gene from the subspecies of carp that you're interested in, you could certainly search for that particular gene. But if you have other ideas on how we could facilitate that, certainly reach out, and we would love to either give some pointers or implement better methods for being able to look at that kind of thing.
We have a couple questions about data formatting. So one person is wondering, is the Great Lakes optimized metadata format available for public use? And then kind of parallel to that, are there published data standards for contributors to make sure that they are optimizing their data?
Good questions. So at the moment, most of the data we've curated internally. So, I mean, we've gone out to these public repositories and done a lot of the curation ourselves. So as more people contribute, this is something we will definitely be improving, is making some of these standards more publicly available. We certainly have them to share, and yeah, I don't believe they're actually on our website at the moment, but we'll put those up shortly. I will say, though, that we are in general following some of the standards that the community has accepted pretty well. So the Genome Standards Consortium has established standards called the MIX-S standards, so the minimum information about any kind of sequence data. So in general, we follow their standards. So if you were following those, it shouldn't require much to convert it to the Glamour formats.
One person is wondering, is all of the raw data held on the SRA, or is it available to download directly from Glamour?
So currently it's our preference to have the original raw sequence data be hosted at NCBI SRA. Sort of as Tim alluded to earlier, we also don't want to be the primary warehouse. And there are downsides to everyone creating their own best new database, then everything gets really fragmented. So we do still think NCBI SRA is the best place to submit the raw sequences. And then we pull from there. And another thing that we're currently working on implementing is the ability to download a lot of the products that we produce, the standardized outputs. So abundance tables or the assemblies from the sequencing data, or the specific gene calls. So those kinds of information are not currently available, but are, again, something that we're actively working on making available for download via Glamour.
Similar to the question for the GLOS folks, is there a future where inland lakes and stream data might be incorporated into Glamour?
So similar to the answer from the GLOS folks, I would say our emphasis has been on the Great Lakes, although currently we're considering the scope of Glamour to be anything in the Great Lakes watershed. And so, for instance, some of the samples that we've identified and have not yet standardized and included in Glamour do come from some of these inland lakes. So if you have some of these data sets and would like to submit them and work with us to get them standardized, there's certainly something that we would be interested in including in Glamour.
All right. Well,
Okay.
I think we can move on then.
Great. And so our third presentation is by Connor Ressler from Great Lakes Commission. He'll be talking about the HABS mapper. So go for it, Connor.
Thank you so much, George. Yeah, thanks everyone for joining today. Like George said, my name is Connor Ressler. I'm a program specialist here with the Great Lakes Commission, and I'll be wrapping up our talks today with a brief showcase of a product from the Great Lakes HABS Collaborative. And this product really is to connect researchers. So in particular, we'll be looking at the HABS Research Mapper for the Great Lakes region. For some brief background, the Great Lakes HABS Collaborative was established in 2015 by the Great Lakes Commission and the USGS Great Lakes Science Center. The Great Lakes HABS Collaborative seeks to improve communication among scientists and between scientists and decision makers on issues related to harmful algal blooms in the Great Lakes. So we know that there is a lot of innovative HABS research being done across the basin, but not necessarily all of those projects are in contact with each other or connect with each other, even as George noted earlier from the Learn meeting. So with the goal of linking science and management in the region with up-to-date information, the collaborative has created the HABS Collaborative Research Mapper, which was launched in March of 2022. So this web-based mapper was designed to visualize the many HABS-related research projects that are happening throughout the region. In its conception, the mapper sought to capture information about the location, the research focus, and the description for all of those different HABS-related projects. And these could be research projects focused either on the landscape, such as edge of field projects or nitrogen and sediment projects, as well as within water bodies themselves. So like HABS monitoring and HABS detection projects. So here's a screenshot of the mapper as it is today. The collaborative is actively developing a communication strategy to promote this mapper, to really build that connected network of researchers, to understand where other projects are being done, and if collaboration between those researchers could be beneficial. So once you get to this webpage here, you can explore all of the different projects on the mapper, either spatially using the map, you can move around and click on different project points geographically, or topically by searching on the list here on the right. You can search by different project names or species names or different funders and researchers and filter through the list that way. When you select a project from the list by clicking the details button there on the right, you are taken to an individual description of the research and the project details. So these details include things like links and documents to learn more about the project, a list of the project team, the funder for the project, as well as an email address of the project's point of contact. That way you can reach out to the team to really build that connected network, that connected web across the Great Lakes Basin. If you are interested in submitting a project point, please note that there are some criteria for the projects, which is listed here. Notably that research must be either ongoing or was completed after January of 2020, and that it has a relevance to either the ecology, the economy, or the social sphere of the Great Lakes Basin that relates to HABS, which the collaborative defines broadly as cyanobacteria or Cladophora. So on the right, you can also see some of the necessary details to submit a project, which include some of the geographic questions, such as the project location, the state in which your institution is located, as well as the water body of focus, plus some additional project details, which include the project funder, the research team names, the project topic, as well as a short description, and short as in 1,000 words or less, which we often have the biggest problem with people submitting. But I do encourage you to take a look at the HABS Collaborative webpage. This QR code will take you directly to the webpage where you can learn more about the mapper, promote the mapper within your own networks if you think this would be beneficial to kind of build that collaborative network across the basin, or to add your project point if your work has relevancy to HABS in the Great Lakes Basin. So we're also welcoming any feedback in an effort to continuously improve the clarity of this work. So please take a moment and utilize this QR code to access the HABS Collaborative Research Mapper. I'll pause here for a moment in case you're still grabbing out your smartphones to take a look at that QR code. But do feel free to reach out to submit a project point, or if you have any further questions about the HABS Collaborative. I've put both my email address here if you have any questions, or the link to the HABS Collaborative webpage. That link right there will take you to learn more, and the landing page has all the information and the place to submit HABS researcher map project points as well. So a little bit of a different spin on HAB's data in the region, and a little quick for you, but happy to take any questions about the research mapper.
Thank you, Connor. Still have a few gloss questions ready if Jennifer would like to answer those. So I'm going to repeat a couple that dropped into the answered section of the Q&A box so that folks who are listening to this later can get the responses. Someone wondered if there are opportunities for smaller firms to become gloss partners.
Absolutely. Feel free to reach out. We are happy to chat about potential partnerships, either US or Canadian.
Fabulous. And then the other question in there was, what does quality control look like, especially for data that's coming in real time from sensors?
So I've been providing I've provided a link for that question on the GLOS website that describes the IU certification process, and also basic information about federal data standards that we adhere to. And so I would definitely check that out if you're interested. I'd also say we do have our own QA, QC processes that we do. We follow QuarTOD, which is quality assessment frameworks developed by the IU's community, and that's run on real-time data. Not all the parameters are covered under QuarTOD, but the ones that are go through the QuarTOD checks before they are put on Siegel.
Thank you. Another GLOS question was, what is the process for downloading and using GLOS data for secondary analysis, or is GLOS more a way to sort of refer researchers back to the people who have the data?
The answer to the asker is both. So you can download a variety of different formats directly from Siegel, CSV, other tabular data, or the raw data. And then we also make that available through an ERDAP service where you can configure really all of the parameters and attributes that you would want to see in the data, and then you download that in a variety of formats as well. So we have a pretty large community of people who do post-processing or analysis on the data that they download from Siegel. And you can also access the data at the API level. We've got a few communities, I guess, that are interested in doing that as well.
Excellent. There's one more GLOS specific question, and then a couple of questions that kind of cover all of these sources. So the GLOS question, is GLOS providing any support for ship flow-through systems? And the question asker mentions an NSF supported project called Rolling Deck to Repository.
So I think that ship flow-through systems is an area that we're looking to grow into. It's not necessarily something that we have a tremendous amount of support for at this time.
Here's a question kind of across the map. Have there been any specific mechanisms for gathering or integrating feedback about all of these platforms, specific feedback from perhaps decision makers or non-research users of the data to make sure that they're as useful as they can be?
So I mentioned that for Glamour, we just recently hosted a workshop for especially the research community, where we also had a lot of help from the Sigler stakeholder engagement team. And we're working to incorporate a lot of the feedback that we got there. And then, so I know the question was specifically about non-research users and decision makers. We're planning on following up with a similar workshop in the future to get feedback from other groups to make sure that we're designing Glamour to be approachable and usable by a wide range of audiences.
As far as the HABS collaborative, the collaborative is a multidisciplinary group on the steering committee. So we are always working and striving to make sure that all of our products are accessible, especially for decision makers or non-researchers. And so we're taking a lot of feedback. If you have any feedback, definitely feel free to reach out and let us know about how the Mapper or any other HABS collaborative products are being perceived. And we always have the mission of connecting all of this amazing research to decision makers, including we have events going on, such as a workshop coming up in April, focused on science and policy in the HABS space for the Great Lakes. And so we're always looking for feedback as we take our products towards that as well.
So I guess I can start and perhaps Tim has more feedback. Gloss does actively engage with its users and since Seagull's been released, we've surveyed our Seagull users a couple of times. One of the very interesting things that we found out with our first Seagull survey is that 30% of our users are researcher manager oriented and 70% of our users are public users. So folks who are just trying to figure out if it's a good day to get off the dock or go fishing. So and that's really great for us, and I think it's something that we're looking at opportunities to re-engage with Seagull users as we move forward to make sure that whatever we're doing and however we're growing, we are giving the people what they want, as they say. Tim, anything else to add to that?
I don't think so. That's a great answer. I will just maybe reinforce that one of our aspirations is, as you said earlier in your presentation, is to be a discovery utility. And so that's, as I said, not necessarily meaning that Gloss houses the data as the data warehouse, but we're able to direct people, whether that's an underserved community or someone like my mom or a researcher or a water intake manager. We're able to direct people where that data is. And that could be through an ERDAP server, it could be through a download, it could be through visualization or even a PDF report that you can download. So I think that's keeping the focus on serving a variety of different user communities will allow us to grow and scale the platform to meet hopefully everybody's needs.
Thank you. I've got one more question from the chat about a different sort of data type. How can the public access digital terrain models produced by LiDAR technology in the region? Is that something that any of these databases would support?
Head, Tim. At present, I don't think we support that type of thing.
That is correct. So the best way to access elevation model data is through the federal government's NCEI. There's a pretty good web application where you can go and query all sorts of data from U.S. Army Corps of Engineers, who are the primary collectors of LiDAR data in the Great Lakes, Office of Coast Survey, and other NOAA groups that collect bathymetric data. It's all available through NCEI, and topographic data is also available through NCEI. Gloss has plans to support elevation models and imagery-based or raster-based products in the future, but it's not currently in our product right now. NCEI is a good resource. That's only on the US side, though. There's not a Canadian equivalent.
I guess one more summary question in the chat before George wraps up. Is there a catalog of all databases like these so we can get a sense of what's already out there?
I guess, this is George. I guess I'd like to answer that. I'm not sure there is. I think it would be really good if Learn had a page where all the repositories were just on one page with a brief descriptor of each. And so I think that should be something that Learn should aspire to get up and running soon as a one-stop shop for new investigators. All right, well, first of all, thank you all for attending. It looks like we had a really good attendance. It maxed out about 180 people. So that really exceeds my expectations. And I'm just thrilled that we could provide this today. And then very special thanks to our three presenters who did a wonderful job describing what the capabilities of each of their repositories provide. And given the number of folks that have logged on, and many may be new investigators, what I'd like to emphasize the importance of the HAB mapper and the Learn directory as tools for early career researchers to establish new collaborations and networking through these tools. So please use the resources that have been described today. Use Learn as a way of meeting folks within the region, and it's been a great event, and thanks also to Sylvia for helping organize and pulling this together. And it's been a great day. So everybody have a good afternoon and go forth. Thanks.
Thanks everyone for joining us.